Hi and welcome to Lessons from the Grand Rounds. In this short video, we'll be learning about some historical points and basics of pathophysiology of ITP. So before 1950s, ITP was termed as idiopathic thrombocytopenia with an unclear pathophysiology, and after the experiments and the study by Dr. William Harrington in 1951, we had some clue about the pathophysiology of ITP, uh, which was established that there is a blood factor in these patients of ITP which is leading to low platelets. So what exactly happened in 1951? So Dr. William Harrington was a hematology fellow at that time, and he was working on patients with ITP. So he hypothesized that there is a blood factor in the patients of ITP which is leading to low platelets, bleeding manifestation, and further complications. So first he himself got infused with the blood of ITP patients, which led to low platelets, bleeding manifestation, and also a convulsion. And then the serial platelets were monitored, and after 4 to 5 days, the platelet recovered. This was further reciprocated to the volunteers of the hospital staff, which where the similar results were found, and they said that there is a blood factor, there is a factor in the blood of these patients with an autoimmune mechanism which leads to platelet destruction. So as we know that there are two major mechanisms of low platelets, or the mechanisms in ITP. The first one is platelet destruction, and the second thing is the platelet underproduction. So to learn about what the pathophysiology of platelet destruction is, let's go with the flow of historical events. So Dr. William Harrington said that there is a blood factor in these patients of ITP which is responsible for these events, and later we found out that these are actually autoantibodies, and the serial specific assays which were developed found out that these autoantibodies are against the platelet glycoprotein receptors, which are glycoprotein 2b3a and glycoprotein 1b9. When these tests were seen in overall patients, we found out that these tests specific to these two glycoprotein receptor antibodies are has very low sensitivity, because there was other specific auto antibodies in the ITP patients apart from only these two glycoprotein receptors. So what exactly happens which leads to platelet destruction in patients with ITP? So assuming we have a platelet here, and there are autoantibodies which tag along the glycoprotein receptors on the platelet. So now these autoantibody tagged glycoprotein receptor based platelet is roaming around in the body, and then it goes to the spleen. In the spleen there are phagocytic cells which has FC receptor, and this FC receptor get attached to these autoantibody tagged platelets, and then these platelets are engulfed by the phagocytic cells, and then they are processed into the phagocytic cell, and the peptides are distributed among themselves and then presented to the T cells, which produce antibodies and memory cells against these peptides of platelet, which further lead to platelet destruction. So this is one of the major mechanisms and the basis of therapeutic options in patients with ITP. The other thing is that these glycoprotein receptors are also present on the megakaryocytes in the bone marrow. So the autoantibodies will also damage the megakaryocytes, which will lead to platelet underproduction. When I said that the peptides of the platelets are being presented to the T cells, and then the antibodies are antibodies are produced, this mechanism is known as epitope spreading, and the final act which leads to the destruction of platelets is either complement activation or apoptosis of the platelets. This was the basis of pathophysiology of ITP. If you have any questions, you can put down in the comments.